Hello. The students who have joined, please write in the chat box if my voice is clear and my screen is visible. Kindly somebody can confirm in the chat box if my voice is clear and the presentation is visible to you. We will wait for 5 minutes and then we will start today's session. Okay, thank you so much. Now we will wait for 5 minutes and then we will start today's lecture. So let's wait for few more minutes so that more students join and then we will start today's lecture. Sorry. So if at any point my screen or my voice is not clear or the screen is not visible, please do write in the chat box. Okay, so let me introduce myself first. So good evening to all of the students who have joined. My name is Moshumi Deb and I am a PMRF fellow in the Institute of IIT ISM Dhanbad. I am a PMRF fellow from the Department of Chemistry and Chemical Biology, and it is a week 6 live interaction session for the NPTEL course of Organic Chemistry in Biology and Drug Development. Just like every session, today in today's session also we will solve some 10 to 15 questions, 10 to 15 MCQs, and these kind of questions will help you to understand the week 6 course material better, and also it will help you to solve the week 6 course assignment which is given to you. And we will start shortly today's session. Let some more students join, and if at any point of today's lecture you have any doubt, please do write in the chat box. At the end of the session I will answer those doubts, and also if you have any doubts regarding today's session, after the session ends you can write in the discussion forum, or also I will just share my email ID so that you can just mail me about the doubt. The today's class lecture and this lecture PPT also will be shared to you by the NPTEL site, and so you can go through. If at any point of lecture you miss something, you can go through this video again, and the PPT also will be available to you. So I think it is 7 p.m. already, so let's just wait for a few more moments, then we can start today's session. Okay. So basically the week 6 course material was concentrated on the oligonucleotide synthesis and the central dogma of the biology. So, mainly this all these courses are related they are just interlinked between the chemistry and biology, the chemistry behind the biological processes of the DNA and RNA, and the translation transcription processes, and also the nucleotide synthesis. So, we will go through the course materials, some related questions, 15 questions today, and let us start today's session. If the students join eventually, we will also add them to this meeting. Okay. So, let us start presentation. Okay. So, the first problem, it says that which of the following statements is true regarding DNA synthesis, and the options are here, like number A, enzymatically it takes place from 3 prime to 5 prime direction only. B, enzymatically it takes place from 5 prime to 3 prime direction only. Option C is chemically it takes place from 3 prime to 5 prime direction, and option D, it says that chemically it takes place from 3 prime to 5 prime direction only. So, when doing this true false kind of question, please read the question carefully, because these questions are sometimes they are tricky, because we read the question wrong, whether it is asking for the true or the false. So, there are several methods of DNA synthesis. Enzymatically is one and chemically is other, and basically this takes place from a particular direction. So, I will first share you the answer of this question, and then we will discuss it for a while. So, it is basically very easy, easy and very I mean it is very general question. So, the answer of this question is, the true statements among these four are number B, enzymatically it takes place from 5 prime to 3 prime direction only. So, there are two methods of DNA synthesis. One is enzymatic and another one is chemical. Okay, somebody wants to join. Let me just admit. Okay. So, there are basically two processes by which we can synthesis the DNA. One is enzymatic and another one is chemical. So, the enzymatic process, it takes place only from 3 prime to 5 prime direction, and in case of chemical it can take place both in the enzymatic and the it can both take place from 3 prime to 5 prime and also the 5 prime to 3 prime direction. So, the answer of this among the true statement among these four will be number B, enzymatically the DNA synthesis takes place from 5 prime to 3 prime direction only. Okay. So, at any point of this again I am saying that if you have any doubt regarding to any question, please write in the chat box. At the end of the session I will answer those questions. Today is the week 6 live session, and we will solve some 15 questions regarding the week 6 class material. These kinds of MCQs will help you to do this week's assignment and to understand this course better. So, now we will move toward the next problem. So, now the problem 2, it says that, okay, I think there is somebody wants to say. Okay, the question is if we do this PPTs will sufficient in NPTEL exam. So, I think this doing only this PPT, it will not be sufficient for your NPTEL exam. I will suggest that these PPTs will guide you that what kind of questions you can expect in the examinations. I cannot say that these questions will be the PPTs are sufficient and only reading this PPT will do. But actually if you do this kind of PPTs and this kind of MCQs, you will understand that how kind of question you will have in your assignments. Okay. Okay, now okay Aurora, I will just answer this question at the end of the session, because let me just finish all the questions and then I will answer your queries. Okay. Okay, thank you so much. Now I will move toward the next problem. So, the problem 2, it says that which of the following statements is false regarding the solid state DNA synthesis. Now, the options are number A, starting point is 3 prime OH and it is attached with silica. Option B is 5 prime OH is protected with DMT group. The DMT, it is the full form of DMT is dimethoxytretyl group, and it is a protecting group for the OH or alcoholic groups. Option C is adenine and thymine are needed to be protected only, and option D is guanine is protected as isobutyryl. So again I am saying, please read the question carefully if it is asking for the true or the false statement. So here in the previous question we were asking for the true statement among the 4, and now we are asking for the false statements regarding the solid state DNA synthesis. I will show you first the answer and then we will discuss the answer. And the correct answer, the false statements among these 4 is number C. Adenine and thymine are needed to be protected only. So why it is the false? Actually the thymine does not need any protection since it does not have any free NH2 group. So why you will need some protection? The protection of any functional group is needed when it is present, right? So if there is no NH2 group, I mean the bases, the main group is the nitrogenous base and it will, the main group is the amine group, and in this group it needed to be protected. And if you do not have this amine group, you do not need to use any protection. So since the thymine, it does not contain any free NH2 group, it does not need to be protected. But the other 3 nitrogenous bases, the adenine and the guanine and the cytosine, they have a free NH2 group. As you can see, adenine have a free NH2 group at this point, guanine has a free NH2 group at this point, and the cytosine has a free NH2 group at this point. So these 3 nucleobases, they are needed to be protected during the solid state DNA synthesis. But the thymine, you can see the structure of thymine, it does not contain any free NH2 group. So thymine does not need any protection. So the false statement among these 3 are adenine and thymine are need to be protected only. Actually the adenine, guanine and cytosine, these 3 are needed to be protected. Adenine and cytosine, these are protected via benzoyl protection group, and the guanine, it is protected by isobutyryl protection group. So I hope I am clear in this problem, and if you have any doubt regarding this problem or any problem, please write in the chat box at the end of the session. I will surely try to answer your query. And if you have still any doubt, you can always mail me and write in the discussion forum. So now we will, I hope this answer and this problem is clear. Now I will move toward the next problem. Okay. Now the problem 3, it says that the correct order of the steps of solid state oligonucleotide synthesis is number A, deprotection of DMT group, coupling, deprotection of base. Number B, coupling, oxidation of phosphorus and deprotection of DMT group. Option C, deprotection of DMT group, coupling, oxidation of phosphorus. And option D is the deprotection of DMT group, oxidation of phosphorus and coupling. So to answer this question, you need to understand the solid phase oligonucleotide synthesis, the steps, how it is proceeding. I mean what is the step which is happening first and what is happening the next. I will show you first the answer, then we will discuss the solid state oligonucleotide synthesis. So the correct order of the steps of the solid state solid phase oligonucleotide synthesis is number C, the deprotection of DMT group, coupling and the oxidation of phosphorus. This DMT, it stands for dimethoxytretyl and it is a protection group for alcohol. Okay. Now I will show you. So this is the cycle of the solid state oligonucleotide synthesis. So at the first you have this kind of a nucleotide. Okay. So here is a base and here is your pentose sugar, and this is the silica, the 3' OH, it is binding to with the silica which is the solid phase actually in the solid phase oligonucleotide synthesis, and this is the 5' OH which is protected via DMT group. So at the first step what happens that the deprotection of this DMT group is happening. So the step 1 is the deprotection. Now how the deprotection is happening? By using acids. Acid catalyzed removal of DMT allows the subsequent base addition. So when we add acid to this protected alcoholic group, this protection breaks and we will have the free alcoholic group at the 5' region. Okay. So now what happens that we have another group, another nucleotide which has the 3' OH, this group is protected. This 3' side, it is protected, and the 5' also has protected. Okay. And this actually this 3' end, it is a triazole, there is a triazole activator. Okay. And this DMT is called the DMT protected phosphoramidate. This group is called DMT protected phosphoramidate. This group is phosphoramidate group. Now at the second step, this phosphoramidate group, it is added to the deprotected or nucleotide. Now what will happen, that this 5 prime OH, it will couple with this this group, okay, and this 5 prime OH will couple with this 3 prime group, and this is the tetrazole activator, and here it will be connected. This will be removed, and this will we will have a phosphorus and oxygen bond. Now, so the next step is the coupling step. First step is the deprotection of the DMT group, and the second step is the coupling of the two nucleotides. And what is the next step? The next step is the step 3 and 4. There are two steps actually, and these two steps which is called together is the capping step. So what happens here, the unreacted OH prime is acylated to prevent further chain extension, and then the phospho ester to it is oxidated to the phosphate by the aqueous iodine. Okay. So the third step is the oxidation of phosphorus. As you can see that there is a the phosphorus here, it is in the 3 oxidation state. There is a 3 bond with the 3 PO bond, and so the oxidation state of this phosphorus is 3. And the third step, what happens, that we add aqueous iodine, and this phosphorus is further oxidized to the 5 oxidation state. Now you can see there are 5 bonds with the oxygen. There is 3 single bond and 1 double bond. So the oxidation state of this phosphorus at this step, it is the 5 oxidation state. And what happens, that its step this stage, what happens, the third step is the oxidation of phosphorus, and now this cycle repeats, and we will have the oligonucleotide. The oligo means 2 to 10. So up to 10 it will this step will just repeat, and at the after that the termination will happen. So the correct order of the solid state oligonucleotide synthesis, first is the DMT group deprotection, the second is the coupling with another DMT protected phosphoramidite, and the third step is the oxidation of this phosphate. So, okay, so by this process 2 nucleotides are joining together, and this cycle will be repeat. Now in the next cycle this DMT group will be deprotected, and again we will add another, and then there will be another nucleotide. So we will have a trinucleotide. So this cycle repeats, and that is how the solid phase oligonucleotide synthesis is happening. Why it is solid state? Because we are using a silica supporting bed to add with the silica this nucleotide is joining, and we are adding 1 by 1 nucleotide at the top of this nucleotide. So I hope it is clear, and if you have doubt, please write in the chat box. At the end I will answer those query. Okay, so I think we can move toward our next question. Okay. So problem 4, it is saying that which of the following figures represent the correct analysis of result of the following experiment. The colonies of E. coli grown in 15 NH4Cl. Okay, here there is a typographical error. This 15 it is represent, it will be like this, it will be in the superscript, like it will be like this 15N. 15N mean it is the heavy nitrogen. This 15 it is suggesting that the atomic mass of the nitrogen, and this 14 it is suggesting the atomic mass of nitrogen. So this will be like this 14 and the 15 nitrogen. Okay, this is the normal isotope of the nitrogen, the mass of 14, and this is the heavier isotope of nitrogen. Okay, this is less abundant in the nature, and it is the more abundant. So the question is, which of the following figures represent the correct analysis of the result of the following experiment. The colonies of E. coli grown in 15 NH4Cl medium are transferred to a medium having 14 NH4Cl as nitrogen source. The DNA isolated after 2 cycles of the growth were subjected to density gradient centrifugation. Okay. So what happening, we are using the colonies of E. coli, and they are growing in a heavier nitrogen containing NH4Cl medium. Now when they will transfer to a lighter nitrogen source containing NH4Cl medium, so and after that the DNA isolated, they are undergoing the 2 cycles of centrifugation, and we will have these kinds of bands we can see in the centrifuge tube. So which will be the correct representation of this experiment? So I will show you first the answer, then we will discuss this problem. So the correct option is the D, number D. After experiment, the centrifugation, it will look something like this. So what it is all about? So let us discuss this thing. Okay, I will just remove this, erase this part. Okay. So Actually, in this case what is happening, like when we have the growth media and it has heavy nitrogen, so we will have a heavy DNA. Heavy DNA means we all know that DNA contain nucleotide and it has a nitrogenous base. Okay. So if the culture medium, if it has heavy nitrogen, like in the red it is representing as 15 NH4Cl. So if it is have 15N, so what will happen, the DNA formed will have a heavy DNA, means the nitrogens will be of this heavy isotope of N15. Okay, and it is representing as red one. And the blue one, the culture medium, it has NH4Cl containing lighter isotope of nitrogen, which is nitrogen 14. Okay. So when the growth media has the heavier nitrogen, the synthesized DNA, it will only have the heavy nitrogen containing, which is represented at red. But what happen when we will be transferring it to the lighter nitrogen containing medium? So now what can be happen? There are three possibilities. Okay, and these possibilities are called conservative. So what the conservative means, this culture medium, it has only this red DNA. Red DNA, it means that only heavy isotope, heavy nitrogen containing DNA. And the lighter medium, it will have similar blue DNA. Blue DNA means only lighter nitrogen containing DNA fragments. So in conservative, what is happening, that the original and new DNA, they do not get mixed. Okay. So we will have a strand which contain only 14 nitrogen bases, and we will have a strand which contain only the red one, all means 15 nitrogen bases. We can have a semi-conservative. Semi-conservative means in one DNA fragment, in one DNA, one strand is red, means one strand contains only 15 nitrogen, and another strand which is blue, means another strand that have only the 14 nitrogen. Okay. So this is called semi-conservative, means these two fragments, they are getting mixed, and we have only one is containing heavy and another strand containing lighter nitrogen. And there is one is called distributive. Now what distributive means, the each strand, I mean we all know that DNA is double stranded. So in distributive, what happens, that in each strand we have both the 15 nitrogen and the 14 nitrogen. So what is the difference between the semi-conservative and distributive? In semi-conservative, we have one strand containing only 15 in double helix between two strands. Say, suppose we can say this is a strand number A and it is a strand number B. So strand number A will contain only the 14 nitrogen, and strand number B we will contain only the 15 nitrogen. But in distributive, both the A and B strands, they will contain in the both nitrogen bases, means in A the 15 and 14 nitrogens is present, and in B also the 15 and 14 nitrogens is present. So what will happen? So it is the centrifugation tube, and you can see that the 15 nitrogen, it is heavier. Okay. So the heavy, because the band for this heavy nitrogen, it will be at the lower end, and for the 14 nitrogen, the lighter one, it will have the higher end. Okay. So what happened when the first generation is incubation happens, after first generation, we will have a band in the middle range of this 14 and 15. So why it is happening? Because we are getting some semi-conservative or distributive. Okay. In semi-conservative and distributive, we have 50-50 ratio. I mean we have 50 heavy nitrogen and 50 lighter nitrogen. So the band is coming at the in between these two places, I mean the heavy and the lighter. We have this in middle places, because this region contains the DNAs which have 50-50 heavy and lighter nitrogen. But what happens after the 2 cycle? Okay. So after the second cycle generation, so when the semi-conservative and this distributive is all done, in the medium we have only the 14 nitrogen. Okay, the lighter nitrogen. So the DNAs they will now have the conservative sequence, and we will have only the lighter band in the lighter region, because we are transferring the E. coli from the medium to heavier to lighter nitrogen. So after the heavy nitrogen incorporation is done, in the medium we only have this 14 nitrogen or lighter nitrogen. So after the second cycle, we will have only the DNA which contain the lighter nitrogen. So we will have these two bands. So and it is represented by this figure number D. So I think it is clear, and if you have doubt, please write in the chat box, and just it is you can read in your section also. There is very okay, let somebody wants to join. So I think it is an experiment basically. Okay. So I think it is clear why the bands, there are two bands at the first. When we are transferring the E. coli from the heavy medium to lighter medium, we have in the DNA we will have both the lighter nitrogen and heavy nitrogen. So the band at the after the first cycle, it is happening in the just middle of the heavier nitrogen and lighter nitrogen, because we have 50-50 ratio. And when the second generation is happening, the medium only have lighter nitrogen, and we have a band at the 14 nitrogen end. Okay. So now we will move toward the next problem. If you have doubt at any problem at any point, please write in the chat box. Okay. So number 5, it is saying that how many nonsense codons are present in the amino acid coding system, and the options are A 2, option B is 3, option C is 1, and option D is 4. So codons, I think we all know what are the codons. The codons are the basically these are the these are a very we can say it is a kind of signal. So we will have 3 nitrogen bases, and these this 3 pair of this 3 nitrogen bases groups, these are called the codon. There are 4 nitrogen bases like A, and we know there are G, there is U, and there is C. In case of RNA, I am saying U because we all know that the amino acid, it is done by the mRNA, and that is why I am saying U, not the T. And the codons, there are the 3 groups, any 3 combination of these 4 bases, they are called a codon. And there are I will first discuss the answer, and then we will just say what is the nonsense codon and what is all about. So answer is number B 3. So somebody wants to join. Okay. So what is the nonsense codon? So yeah, as you can see that these are all the codons. Okay, these are all the combination possibles by this 4 nitrogenous bases of uracil, cytosine, adenine, and guanine. And now what happens that these all combinations, they are particular for adding one amino acid to the translation or the protein synthesis steps for by the mRNA. Okay, like this UUU and UUC, these are responsible for adding the phenylalanine in the chain. The UUA and UUG, it is necessary for the addition of the leucine, and so these are the codons for responsible for the adding the amino acids. And there are 3 codons, particularly this UAA, UAG, and UGA. So these are called the stop codons or the termination codons. So these codons provide a signal for the termination of polypeptide chain, and these are the UAA, UAG, and UGA. And UAA, it is called ochre, UAG, it is called amber, and UGA, it is also known as opal. And these are called the nonsense codons, since they are not responsible for adding any amino acid to the polypeptide chain. Only they will signal the system that now it is time to stop the polypeptide synthesis. Okay, and it will terminate the chain, the polypeptide chain. And now you can, if you are having this, you are thinking that you have to remember all these codes and all these amino acids. So you don't need to remember these codes and these amino acids, that which code is responsible for which amino acids, but you have to remember 4 codons. Okay, so these 3 are one, this UAA, this UAA, this UAG, and this UGA. These are stop codons. You need to remember what are the stop codons. This UAA, UG, sorry, okay, there is some problem. So you need to remember like these 3 codons, they are stop codons, and another one codon which you need to remember, which is the AUG codon, A, U, and G. So this AUG codon, it is responsible for the addition of the methionine amino acid, and it is called the start codon. So when we will have this combination in our polypeptide in the synthesis, when we will have this combination of this bases is AUG codon, and it is called the start codon. From this end, the amino acid synthesis, the polypeptide chain synthesis starts actually. So it is called the start codon. So, just remember 4 codons, this top 3 stop codon UAA, UAG and UGA, and one start codon, one and only start codon, which is AUG, which is responsible for addition of methionine. So, I hope it is clear, and it is very simple and general question. Just remember this 4 codons and it is enough. You do not need to remember all these codons and all these combinations. Okay. So, now next the problem 6, it says that which of the following statements regarding the ribosome is false. Number A, ribosome of prokaryotes is smaller than eukaryotes. Number B, ribosome is made up of ribosomal RNAs and the proteins. Option C is the S unit of ribosome stand for the Sedberg unit, and option D is the eukaryotic ribosome contains 50 S and 30 S subunits. Again, this is a true false statement, and just read the questions, all the options carefully, and it is very you can just answer this question easily if you this is a very basic question actually about the ribosome, and the ribosome, it is responsible for the polypeptide synthesis, a part of the polypeptide synthesis. And first I will show you the answer. Okay. The answer is number D, the eukaryotic ribosome contains 50 S and 30 S subunits. So, it is the false among these 4 statements. So, this is the basically the prokaryotic ribosome and eukaryotic ribosome. So, as you can see that the prokaryotic ribosome actually it is very smaller than the eukaryotes. Okay. And it is the prokaryotic ribosome, it is called the 70S ribosome, and the eukaryotic ribosome, it is called the 80S ribosome. In case of this prokaryotic ribosome, it has the 50S subunit, this bigger one, and the 30S subunit, this smaller one. Okay. And this eukaryotic ribosome, it has a 60S subunit, which is this bigger subunit, and the 40S subunit, which is a smaller subunit. So, the number D is false, because it says that the eukaryotic ribosome contains 50S and the 30S subunits, but it is the false because actually the prokaryotic ribosome, it contains the 50S subunit and 30S subunit, and the eukaryotic one, it contains the 60S and 40S subunit. Okay. So, I hope it is clear why this number D is the false. Just to remember the subunits of the prokaryote and the eukaryote ribosomes, and otherwise it is a very basic question. And now I will just move toward the next question. Okay. So, the problem 7 is says that the position where the tRNA will bind with the ribosome depends on its codon sequence, intramolecular hydrogen bonding, anticodon sequence, and hairpin like structure. Okay, somebody wants to join. Okay. So, number 7 we were at. So, the position where the tRNA will bind with the ribosome depends on its codon sequence, intramolecular hydrogen bonding, anticodon sequence, and hairpin like structure. So, I will just show you the answer. The answer of this will be anticodon sequence. So, what is the anticodon sequence and what is happening? So, it is the tRNA. Okay. Now, this tRNA, it has the 3 loops. Okay. The first is called the T loop, and there is an anticodon loop and a D loop, and at this 3' position the amino acids are getting attached, and there we have a combination of 3 nitrogenous base on this anticodon loop. Okay. And this combination is called the anticodon combination. So, this is the codon, this is the mRNA, and on the mRNA we have this 3 sequence of this nitrogen base, which is called codon, which we just discussed in the problem number 5. Like codon, and these codons are responsible for the addition of the amino acids. So, what happens, there is a point, there is a in the tRNA that depends on this anticodon, it will bind to the ribosome. Okay. So, the mRNA, it has codons, and this codons, according to this codons, this tRNA we will have a anticodon, and according to that the amino acids will be bind here, and we will have a polypeptide chain based on this thing. Okay. So, just remember that the tRNA, it is the anticodon sequence, it is responsible for binding of tRNA to the ribosome and the synthesis of the polypeptide chains. Okay. So, again I am saying, if at any point of this lecture, at any question, if you have doubt, please write in the chat box. At the end of the session I will try to answer your queries, and also after today's session if you have the doubts, please write in the discussion forum, or you can also write a direct mail to me regarding today's session. If you have any problem, I will share my mail ID at the end of this session. Okay. So, I think it is clear. If I will now move toward the next problem. Okay. The enzyme responsible for removing the introns and joining the exons together is called number A lysozyme, number B ligase, number C spliceozyme, and number D EcoR II. Okay. First I will just show you the answer, then we can discuss this problem, what is the introns and what is the exons, and what is all about. So, the correct answer is spliceosome. The spliceosome enzyme, it is responsible for removing the introns and joining the exons together. Now, all it is about actually the polypeptide synthesis by the mRNA. Okay. Now, what happens, like when the polypeptides are synthesizing, the amino acids are joined by the using of the codons. Like in the previous slides, I was showing you the, we will go to the previous slides where we had discussed that problem. Like, okay. So, as you can see that there are the combination of 3 nitrogenous bases which are called the codons, and these are codes for addition of amino acids in the polypeptide synthesis, and it is happened by the mRNA. Like we have this kind of structures on the codons on the mRNA, and by this structure one by one amino acids are joined, and we have this polypeptide chain. Now, what happens, like we have 4 nitrogenous bases, and we can have total 64 combination of this 3 letter codons. Okay. So, we can have the 64 combination of this 3 nitrogen bases, but we only have 20 amino acids. Okay. So, not all this codons we will be responsible for addition of amino acid. There will be some codons which do not add any amino acid to the chain. Okay. Now, what happens, they are also present on the mRNA as codons. Okay. There is a called intron and exons. So, the exons, it is a combination of a polypeptide chain, and after that if the codons are present which do not incorporate any amino acid, that part will be said intron. So, we have exon and in between we have some introns where no amino acid is incorporated. Okay. All means they have codons, but they are not responsible for addition of any amino acids. So, that part is called intron, and the exon part, it is the polypeptide sites. Okay. The polypeptide sites, the one by one there are polypeptides, and there is some joining of this ends. Now, what is happening, you can see in this figure that we have a exon part here, and then we have a intron part here. This intron part, this does not have any amino acid. Then again we have a exon part, and there is an enzyme called spliceozyme. This spliceozyme, what it will do, that it will just split this intron part. Like it does not have any amino acids, so we do not need it in our polypeptide chain, and this spliceozyme, it will just cut the intron out, and we will have the exons joining together, and we will have a mature RNA. Okay. So, the first, it is at the first the synthesis of this polypeptide, it is not called this is called the basic RNA, it is formed, and then we will have a mature RNA which is responsible for the amino acid translation. So, what the spliceozyme is doing, it is just taking out the introns part which are not responsible for any amino acid addition, and the spliceozyme will just cut those parts, and we will have a combination of all the exon parts which are which contain amino acid and the polypeptides. Okay. So, just remember the name of this enzyme spliceozyme and its role for joining intron and exons. So, I hope it is clear, and we will just now move toward the next problem of today's session. So, again I am saying, please write in the chat box if you have any doubt at any point of session. Okay. Okay. So, the problem 9, it says that the reaction in DNA replication catalyzed by DNA ligase involves A. Addition of new nucleotides in the leading strand. Option B. Addition of new nucleotides in the lagging strand. Option C. Base pairing of the template and newly formed DNA strand. And option D. Formation of phosphodiester bond between 3' of one Okazaki fragment and 5' phosphate of the next lagging strand. So, the DNA replication, it is basically catalyzed by this DNA ligase, and the reaction, the correct statement or the correct answer of this problem is the number D. The formation of the phosphodiester bond between the 3' OH of one Okazaki fragment and the 5' phosphodiester 5' phosphate of next lagging strand. So, this is the DNA replication step. As you can say, we have a double stranded DNA. Okay, here there is a double stranded DNA, and this strands are there is a breakage. Okay, somebody wants to join. So, at first what happens that this double in the DNA replication reaction, at the first there is a helicase enzyme. Okay. Now, what the helicase enzyme does, it just unwinds this double stranded DNAs into the single strands. Okay. So, there are two strands are formed, okay, one is called the leading strand and one is called the lagging strand. And what is happening here, that we have a primase, and this primase, it adds a RNA primer which just starts the synthesizing of the another fragment of this DNA strand, and there actually this DNA replication, it is discontinuous. What is the discontinuous means? The fragments which are formed, they are not I mean there is not continuous. We have the fragments of this DNA strands. Okay. Now, what happens? These fragments are called the Okazaki fragment, and what happens? Like this formation, the DNA ligase, what the DNA ligase does? The DNA ligase just joins these fragments. Okay. Now, what it does? It forms the phosphodiester bond between the 3' OH of one Okazaki fragment and the 5' phosphate of the next lagging strand. Okay. So, just remember that the DNA replication process, it is not continuous one, means we do not have a continuous formation of the strand of the DNA, it is discontinuous, means we have some fragments, and these fragments are called the Okazaki fragments. As you can see in this part, this blue part, it is called this Okazaki fragments, and these are joined together to form a strand of new DNA by the ligase enzyme. So, there are various enzymes actually in the DNA replication, the topoisomerase, there is a helicase, there is a primase, and there is a ligase enzyme. So, it is better to remember the roles of this enzyme in the DNA replication process. So basic, you do not need to remember everything, just have a basic concept about the DNA replication and what is the role of these enzymes. Okay. So, I will move toward the next problem. So, problem number 10, it says that which of the, somebody wants to join. Okay, so problem number 10, it says that which of the following is responsible for the initiation of RNA polymerase activity in bacterial state. The options are A, initiation site, option B is promoter region, option C is sigma factor, and option D is rho factor. And the correct option, it will be answer number C, sigma factor. And I will just let you show, yes. So, what is the sigma factor? It actually binds the code enzyme to convert it to the hollow enzyme. And what is the enzyme? It is the RNA polymerase enzyme. Okay. So, it is encoded by the RPOD gene. There is a gene which is encoding the sigma factor, and it has a critical role in promoter recognition, but it is not required for the transcription elongation. It recognizes the correct promoter site by decreasing the affinity of enzyme to the non-specific DNA sequences, and the amount is only 30% amount of the enzyme. So, what is the sigma factor is doing? Actually, it is helping for the recognition of the promoter site. Okay, the whole the DNA chain, it is not required for the transcription. Thus, there is a certain part of the DNA which is required from where we need to start the transcription process. I mean the formation of the RNA from the DNA, which is called the transcription, and it is for all the DNA, the total DNA strand, it is not required for this transcription. So, for where it is where we need to start the transcription, that is shown by the promoter region. Okay. And this promoter, okay, there is something in the chat box, I think. Okay, no, I Okay. So the promoter region, it is we can we can see like the promoter region, it is specifying like where we need to start the transcription. Okay. So now how this RNA polymerase enzyme, it is actually helping the formation of RNA from a strand of the DNA, from a template strand of the DNA. So how will this RNA polymerase will recognize the promoter strand? So there is a thing called, it is called the part, it is called the sigma factor. Okay. And it binds to the RNA polymerase core enzyme to make it a hollow enzyme, so that it can start the transcription process. And after the promotion, the promoter recognition, this sigma factor, it falls off, and we do not need, after the RNA polymerase it's know that what is the promoter site, it does not need the sigma factor, and it just removes the sigma factor from this place. Okay. So the correct answer will be the sigma factor. Just remember what the sigma factor, name of the sigma factor, and what it does. So it helps in the promoter recognition. So I think I am clear about this problem. Now quickly I will move toward the next problems. Okay. So problem number 11, it says that what would be the complementary RNA strand for the following DNA sequence in the transcription process. The DNA sequence is given as 5 prime GCG TATG 3 prime, and the options are like this. Option A is 5 prime GCG UAUG UAUG 3 prime. Option B is 3 prime CGCA UAC 5 prime. Option C is 5 prime CGC AUAC 3 prime, and option D is 3 prime GCG UAUG 5 prime. So now this problem, it may be a little bit confusing for many people. So I will just show you the answer and then we will discuss. So the correct answer will be number A, 5 prime GCGU AUG 3 prime. Okay. So that what is the transcription process? It is the formation of RNA from a template DNA. Okay. So we all know that the DNA, we have the two strands in DNA, which one goes from the 5 prime to 3 prime, and the another goes from the 3 prime to 5 prime. Okay. So now both the strands are not used for the formation of the RNA from the DNA. One strand, it is used for the formation of RNA, and the another strand, it is not. So the formation of RNA, it happens from the 5 prime to 3 prime direction. Always it happens from the 5 prime and 3 to 3 prime direction. So which strand will be used as a template? The 3 prime to 5 prime strand, which is this lower one in the figure, you can say. Now see, the lower one is the 3 prime to 5 prime strand. Okay. And it is called the template strand or non-coding strand or antisense strand. This strand, it is used for the formation of the RNA. Okay. So if there is a code like there is a T and G and TA G C, as you can see this there is a combination of bases TG TA here. So what happened, the combination of template strand, the bases we have, the RNA, it will add the complementary things. Okay. So since there is a T thymine in the DNA, it will have it will add a adenine for that. It has a G, it will have a cytosine for that. There is a T, it will have a adenine here, and it has a adenine, it will add uracil. In RNA we do not have thymine, so it will be uracil here, and it goes from 5 prime to 3 prime. So and it is called the template strand, means based on this strand we will have this RNA. And the other one, which is not used for the transcription, it is called the non-triplet or the coding strand or the sense strand. So why it is called the sense strand or coding strand? Because as you can see, it is similar, it is I mean completely similar with this synthesized RNA, as the code is ACAT and the sequence is ACAU, only we have uracil in place of thymine, and it is also 5 prime to 3 prime. Okay. So in the question, the DNA sequence which is given to you, it is for the non-triplet or the coding strand. Okay. Okay, I think it will be better if you are given here the coding DNA sequence or coding strand mentioned here, otherwise it is maybe little bit of confusing. So I just will add something, the following coding DNA sequence. So if it is coding is given, okay, I am just saving. Okay. So, if the coding is given, so just remember the RNA sequence, synthesized RNA, it will have the same sequence like the coding strand, just that there will be uracil in place of thymine. So, answer A will be the correct answer for this question. Okay. So, I hope it is clear why it is just remember there are two strands, one is called the template strand or the coding strand or sense strand, and the another one it is called the template strand or non-coding strand or antisense strand. The non-template strand it goes from 5 prime to 3 prime, and the template strand it goes from the 3 prime to 5 prime. This 3 prime to 5 prime template strand it is responsible for the RNA synthesis, and the RNA synthesis is goes from 5 prime to 3 prime again, and based on this template strand the complementary bases will be added, and this synthesized it will have the similar pattern as the non-template strand, only uracil is present at the place of thymine. Okay. So, I am clear. If you have doubt, please write in the chat box, so I can just clear your doubt at the end of the session. So, now we will just move toward our next problem. Okay. So, the problem 12, it says that after unwinding the double helix, the supercoiling of DNA is prevented by the enzyme DNA topoisomerase, DNA ligase, DNA helicase and DNA polymerase. So, the correct answer will be DNA topoisomerase. So, when we were doing that DNA replication question in the previous one slide, I said that there were various enzymes which are taking part in this DNA replication process, okay, and each enzyme has a very specific roles in that process. Okay. So, as you can see there is several enzymes, okay, and these enzymes what are they doing, just remember what is the role of each enzyme. So, the first step in DNA replication is to separate the strands of DNA using the helicase. So, we have an enzyme called helicase, this blue triangle enzyme helicase. What it does, it just breaks the hydrogen bonding between the complementary nitrogenous bases between the two DNA strands. So, you can see there are two DNA strands and there is a helicase enzyme, it is breaking the hydrogen bonding between the each complementary DNA bases and this is the start of the initiation, and this is the single strand binding proteins are present here, and this single strand binding proteins they are preventing these just fragmented DNA parts to just bond with each other again. So, then we have a DNA primase that comes in to create the RNA primer. So, again now then we have another enzyme, it is called the DNA primase enzyme, and what the DNA primase enzyme does, it creates a RNA primer, and this RNA primer what it does, it just add some new bases and together and forms the new strand of DNA. And then we have a DNA polymerase enzyme that comes in and synthesizes that new strand of DNA. So, the RNA primer it just connects some nitrogenous bases, and the DNA polymerase what it does, it just bind those bases together and forms the hydrogen bonds between the two strands, and new strands are forming. So, single strand binding proteins make sure the two strands come back, which I said here. So, now topoisomerase. So, it helps to relieve the tension created by unwinding the DNA. So, actually it is a very general experiment you can do if you have two ropes and you are just twisting two ropes together. Now, when you start to just unwind the ropes, you have two ropes, we are just twisting two ropes together by rotating. Now, when you try to unwind this ropes, what you can see, the place where you are trying to unwind, there is a coiling of the ropes. Okay, you can just try it at your home, it is very easy experiment. Just when you try to unwind the ropes, there at the place of the unwinding, there is the ropes, they will just strangle more closely together. Okay, that is called also super coiling. In case of DNA, the thing is just as same. So, but we need to prevent the super coiling, so that the helicase can do its work and the replication process it can run smoothly. So, topoisomerase enzyme it has the function of relieving this stress and the tension created by the DNA, unwinding the DNA. Okay. So, the question after unwinding double helix supercoiling of DNA is presented by the enzyme topoisomerase. It relieves the tension of this breaking of this hydrogen bonding by the helicase, and this is the role of this topoisomerase. And finally, DNA ligase enzyme, it sticks the Okazaki fragments together in the lagging strand. Okay. So, again I am just repeating what the role of the different enzymes in the DNA replication. Please remember the roles of this different enzyme in the DNA replication, because it is a very important part of your course, and the many questions can be come from this part. Okay. So, of the DNA replication, the first step, that there is a helicase, which is this is separating the two strands of DNA, making it single strands. The single strand binding proteins, they are they will just make sure that the two strands does not come back together again. And the topoisomerase, it is the unwinding the tension of this breaking of these strands. And there is the DNA primase, which is creating the RNA primer. Okay. DNA polymerase, it synthesizes new strands of DNA, and DNA ligase, it strikes DNA polymerase in the leading strand. The it is happen the sticking, it happens between the formation of bonds, it is taking place with the help of DNA polymerase. And again in the lagging strand, it is the DNA ligase which is connecting the Okazaki fragments and forming the new strand of DNA. So, please remember the role of the each enzymes in the DNA replication, it is very important. Okay. So, now we will move toward the number 13 question. It is says that the DNA polymerase 1 in E. coli, Escherichia coli, has specific function of number A, synthesis of leading strand in DNA, number B, synthesis of lagging strand in DNA, number C, covalent linkage of adjacent 3' OH and 5' phosphate groups, and option D is removal of the RNA primers and replacing them with DNA. So, I will quickly show you the answer, then we will just discuss. So, there are 5 types of the DNA polymerase enzymes, number 1, 2, 3, 4 and 5. Okay. So, there are characteristics of DNA polymerase in E. coli. So, in E. coli there are 5 type of DNA polymerase. Okay. The 5' to 3' polymerase activity, it is present in all the 5 DNA polymerases. Number the 3' to 5' exonuclease activity, it is present in only number 1, 2 and 3 DNA polymerase, not in 4 and 5. And the 5' to 3' exonuclease activity, it is present only in DNA polymerase 1, not in the other 4. And what is the function of this? Just if you do not remember this part, it is also alright, but please remember what is the function of this each of this 5 DNA polymerases in the E. coli bacteria. Okay. So, in the DNA polymerase 1, what is its function? It removes and replaces the primers. What is the primers which we discussed just in the previous slides? What is the RNA primer? Sorry. So, DNA polymerase 1, what it does, that it removes and replaces the primers. DNA polymerase 2, it rolls the DNA repair, restarts the replication after damaged DNA, halts the synthesis. DNA polymerase 3, it elongates the DNA. Okay. And DNA polymerase 4, it role is the DNA repair, and DNA polymerase 5, the role is DNA repair and translation DNA synthesis. So, the question is asking for the role of the DNA polymerase 1, and the correct statement will be number D, removal of RNA primers and replacing them with DNA. Okay. So, this one removes and replaces primer. So, just remember the function, this 5 function of this 5 DNA polymerases in the bacteria E. coli. So, that is enough. If you can remember this part, it is very good. Otherwise, remembering only the functions is also fine. Okay. So, now quickly I will move toward the, we only have 2 questions remaining. So, number 14, the question asks that which of the following statement regarding RNA is false. Number A, rRNA acts as a catalyst to make the peptide bond. Option B, the amino acid sequence of proteins depend on the base sequence of the mRNA. Option C, RNA polymerase 3 synthesizes the mRNA, and option D is RNA polymerase 1 synthesizes the rRNA. So, I will quickly show you the answer because we have very few time left for today's class. So, the correct answer of this problem will be answer number C, RNA polymerase 3 synthesizes mRNA. It is the false statement. Again, read the question carefully, it is asking for the false statement, and number C, it is the false statement. So, Like DNA polymerase, we have 3 type of RNA polymerase. We have RNA polymerase 1, RNA polymerase 2 and RNA polymerase 3. And in this table you can see that there is a very beautiful comparison of these 3 RNAs. So, remember RNA polymerase 1, it only transcribes ribosomal RNA or rRNA. Okay. So, RNA polymerase 1, it synthesizes rRNA. What is the role of RNA polymerase 2? The central enzyme that catalyzes the DNA directed mRNA synthesis. So, the RNA polymerase 2, it synthesizes mRNA, and what the rRNA does, RNA polymerase 3 does, it transcribes small untranslated RNAs such as tRNAs. Okay. So, RNA polymerase 1 synthesizes rRNA, RNA polymerase 2, it synthesizes mRNA, and RNA polymerase 3, it synthesizes the tRNA. And there are other comparisons like you can just read from this table. So, just remember this only the first part, only the first 3 things, like which RNA polymerase is responsible for synthesizing which RNA. So, RNA polymerase 1, it synthesizes rRNA, RNA polymerase 2, it synthesizes mRNA, and RNA polymerase 3, it synthesizes tRNA. So, the false statement will be number C. So, just remembering only this first part, first row is enough for this RNA polymerases. And the last question of today's session is the recognition sequence of the Pribnow box -10 is there are 3 prime TA TA AT 5 prime, number B is 3 prime TT GA CA 5 prime, number C 5 prime TA TA AT 3 prime, and number D 5 prime TT GA CA 3 prime. And the correct answer is number C 5 prime TA TA AT 3 prime. So, okay. So, just a bit before some time we just discussed about the promoters and RNA polymerase, that how sigma factor it helps RNA polymerase to know what is the promoter site, and the promoters are the specific region in DNA that are recognized by RNA polymerase, and this recognition is helped by the sigma factor. Okay. So, the start site it is designated as the +1, and it is the first base that will be copied to the RNA, and the -10 sequence, it is called the Pribnow box sequence, which is a sequence that contained within all prokaryotic promoter region. Okay. So, the promoter region, it contains this -10, this sequence, the TAAT T, and it starts from the 5 prime and goes to the 3 prime, and the sigma subunit, the -35 sequence, it is the second recognition site, and the sigma subunit recognizes the sequence and connects to with it. The RNA polymerase then comes and in contact with the Pribnow box, and then it can start the synthesizing the RNA from the DNA template. So, it is the diagram of the promoter region. The RNA polymerase does not copy the promoter sequence into mRNA, and because the promoter is to the right of the transcription start point, is to the right of the promoter. RNA polymerase cannot copy another promoter sequence. Minus consent sequence is copied but into DNA, not into RNA. Promoter is copied and processed and spliced into mRNA. So, just remember what is the it is not a very important thing to know the every concept of this Pribnow box. Just remember the sequence. Okay. What is the sequence for the -10 region? It is the TATAAT, and for the -35 region it is TTGACA. Okay. So, this was the last question of today's session, and again if you have any doubt, okay, yes, against the query, मैम नाउ आंसर प्लीज़ क्वेरीज लाइक मैकेनिज्म भी करने हैं क्या एग्जाम पॉइंट ऑफ व्यू से, MCQ आता है ना. ओके. सो अरोड़ा, आई विल जस्ट say that I am doing only, I am the TA for your session only for the assignment part. Okay. There are another TAs, they just conduct the I just look over the assignment part. So, for the main examination I think there won't be only the MCQs. In the main examination you will have the broad questions. So it is better to remember and understand the mechanism and prepared by that. And also I will suggest you, please put your query in the discussion forum of the NPTEL. Okay. So the main question and the final questions, they are set by other TAs, and I am not TA for the your annual, the final exam. I am only TA for your this assignment and MCQs. So doing this kinds of MCQ, okay, contact number, no, sorry, I don't have any contact number, but there is a discussion forum of NPTEL. You can see that. Okay, I will just show you. If you are saying that online, okay, so in the NPTEL website, okay, so I will just sign in. So in the NPTEL website, okay, one second, I will just go to the, I will just quickly go to your online courses of NPTEL site. Okay. So here, yeah, here you can see, I think my screen is visible, and you can see there is a ask a question option here. Okay. So when you click on this ask a question option, you will be guided to a page, or there is a discussion forum. Okay, and this in that discussion forum you can write a mail. That email site, there is a okay, there is something, I think, error. So there is a Google group, and you can put your email at this side. Okay, you can see there is a Google group. When you add the AXA discussion, you just enter this, join this discussion forum, and you can write a conversation. Okay, you can write an email. That email will be go to the supervisor, the professor, and the official TA of this course, and they can answer this question. But I think for the final examination, you will have to prepare the not only the MCQs, you have to prepare for the broad question. But yes, this anything you can ask there, and for on the assignments, only doing the MCQs is fine, because in assignments you only MCQs will be asked. Yes, I will suggest that you should prepare for the mechanisms. Okay, not the detail, but just concepts you need to be know, because in the final examination they can ask you the mechanisms. Okay, and if you have any doubt, paper ma'am MCQ होगा क्या? So I don't know about the final paper. Assignments are only MCQ, but the final paper, it will be it will contain the broad question. It will not have only MCQ. It may have some MCQs. Okay, it may have some MCQ, but it will not have only MCQ. They will have the broad question or MSQ kind of thing, fill in the blanks kind of thing also. So I will suggest just do this. Go to this NPTEL website and just join this discussion forum and write an email, and then you will have the proper answer for how the examinations will be conducted and anything. And if you have any doubt about today's session, please write an email to me, or you can also write in this discussion forum. I will just share my email ID with you. Okay. So this is my email ID, and if regarding to this session you have any doubt about the MCQs and what we learned today, so please write an email. And otherwise, if you have doubts for the final examination, please write in this discussion forum. Just join this discussion forum of organic chemistry and biology and drug development. The link is present in your NPTEL website, and there we will you can just write email, and the course instructor, the professor, they will answer your query. Okay. So I think we can close today's session, and thank you all for joining, and we just we will meet you next Friday for the next week 7 course material. And thank you all for joining today's session. Now I will close today's meeting. Okay, thank you. Okay, thank you all.